Hi, welcome to this module. This is a short module. So, and it will be focused more on the electrodes that are there for measuring or acquiring the EEG signals. Okay. So, after this module we will see the application of the microfabrication for several devices just as an example. So, if you see this slide, what you see here are the EEG cup electrodes from MVAP. This is a company, it is a medical supplies and corporation in California, and there are several kinds of electrodes that are used for capturing the EEG signal. The first one is a cast style gold cup electrodes. We will show it to you how these electrodes looks like. We will bring it and show it to you. There are stamped style heavy duty wire silver cup electrodes which are generally used, and then there are stem type heavy duty wire gold cup electrodes which is right over here, cast style gold cup electrodes which is here. We have insulated gold cup electrodes, then we have insulated silver cup electrodes, and so on and so forth. This one are the dry electrodes. So, these are all wet electrodes, wet electrodes. All right. What are wet electrodes? Wet electrodes requires a gel to be used for before you can place it or stick it on the scalp. This schematic shows dry electrodes. Dry electrodes does not require a gel for making a contact to the scalp. Now, what is the importance of using the gel? The importance of using the gel is to reduce the impedance. All right. In this case, you can see there are spikes, and thus the chances of touching the scalp is higher without using any gel. All right. We will discuss this thing in detail. Now, what are the applications of this kind of EEG electrodes? One of the application is neonatal hearing screening. When the baby is born, to understand whether the baby can hear it correctly or not, there are several techniques commercially available. One is called otoacoustic emission, which is the echo generated by hair cells in cochlea when presented to the auditory stimulus, and OAE, which is a short form of otoacoustic emission, reflects the status of cochlear amplification in the auditory pathway, while the another technique is called auditory brainstem response, or ABR, is a far field summon potential captured at scalp areas, you can see here, which reflects neuron activities from cochlea to brainstem within 10 microseconds. So, typically when you talk about ECG versus EEG, we will be discussing this thing in detail. You will see that the voltage or amplitude of the signal in ECG is millivolt, amplitude of signals in EEG is microvolts. You can see here typical ABR amplitude is 0.1 microvolts to 1 microvolt, and thus your electronic module should be strong enough with a higher signal to noise ratio, such the signals can be amplified and the noise can be filtered out. All right. So, we will be discussing this in detail. We will discussing about auditory evoke potentials. That means that the ABR technology test entire hearing pathway from the ear to and including the brainstem. So, if you start from the ear, it goes to the brainstem. All right. And OAE test a portion of the hearing pathway from outer ear to the inner cochlea. That means, if you see here the blue color line, OAE otoacoustic emission starts from the outer ear to the cochlea, while ABR starts from the outer ear all the way to brainstem. But what we are interested in? We are interested in not just to brainstem, but all the way to auditory cortex, starting from here, right, all the way to here. And how can we do that? What are the ways to do that? We will be discussing. All right. This is an important area of research where you can understand that how you can use these EEG electrodes to understand whether the hearing is correct or not, and also to understand the cognitive ability of the newborn. So, we will be discussing about several signals including mismatch negativity, ABR, what are the positive peaks, what are the negative peaks, what is P300 positive peak at 300 milliseconds, and so on and so forth in the modules. However, this particular class is more on understanding just a general concept about the EEG and the fabrication techniques for the same. So, when you talk about EEG placement, you see this is the way they use, which is called 10-20 system for placing the EEG electrodes, and then you can see that the numbering system on the system is odd for the left side and is even for the right side. What do you mean by odd for the left side? You see this side, if I show you this side is a left side, alright, and the remaining is the right side. So, if you see the numbering 1, 7, 3, 3, 3, 1, 5, 3, 1, this is all left side, that is why it is odd. It is just to help in understanding how to place the electrodes, alright, and how to use the numbering system, while the right side all the numbers are even 2, 4, 8, 4, 4, 2, 4, 6, 2, right, while the regions, brain regions are divided or are noted in following ways. The F, which is the alphabet F, is used for frontal area, alphabet T. So, you can see here frontal area like this, alright. T is used for temporal, T here, okay. O is used for occipital, O, occipital, alright. So, the idea here is that 10-20 system is an internationally recognized method. It ensures inter-electrode spacing is equal, right, because you can see that how it should be placed, because for different skull size and shape we need to make sure that the electrode placement is correct, and it covers all the brain regions, right, from frontal, temporal, occipital. When you see the brain, right, you cannot really identify what is the difference, but when you look at the anatomy, right, we kind of know that what are the regions required for motor cortex, what are the regions that are used for your visual stimulus, what are the regions that can be which helps in understanding the hearing of the things. The idea is that this electrical signals travels on entire brain surface, right, but at some point of time or in some cases the electrical signal starts misfiring. I am putting in very easy terms, okay. So, if that happens, then it is kind of epilepsy, and we talk about epilepsy and how can we record those signals from the brain. There is a device that we will fabricate, right. So, let us see that the other devices that we can fabricate with microfabrication, and then we will take care of what is the difference between ECG and EEG, followed by the experiments related to the understanding the EEG signal, acquiring the EEG signal, and also understanding how we can use the MATLAB to solve or to process these signals further. Yeah. All right. So, till then you take care. I will see you in the next module.